Hello everyone, my name is Manny the Microscope, and I'm here to welcome you to a presentation on Confocal Fluorescence Microscopy. To help me explain this type of microscopy, I have my three friends. Molly, the Queen of Microscopy, who will be explaining the principle of Confocal Fluorescence Microscopy. Barry, the Bacterial Cell, who will be discussing the advantages of using this type of microscopy over others. And last but not least, Gamma Venin, who will be giving an example of the application of this technique. Well, alrighty then, here we go.
Greetings, my fellow science explorers. My name is Molly, Queen of the Microscope, and I'm here to explain the principle of confocal fluorescent microscopy. The confocal microscope is a specific fluorescent microscope that allows for the production of 3D images with high resolution. Compared to a wide field microscope, the confocal microscope contains two major differences. The first being a laser, which is the main light source. There is also something known as a pinhole. There are two, one located in front of the laser and another in front of the detector. The detector is also known as a digital camera source where images are being produced. Both pinholes help in the reduction of out-of-focus light to improve image quality. To start off the process of how the image is being produced, we must begin with the laser. The light of the laser will enter, bouncing off the dichroic mirror and rotating mirrors, focusing on one defined spot of the specimen. This is known as the excitation light. The fluorescent dye on the specimen will give off fluorescence that will travel towards the digital camera, bouncing off the rotating mirrors once again, and transmitting through the dichroic mirror. This is known as the emission light. The next stop is the pinhole. The pinhole in front of the detector will block the out-of-focus light, not allowing it to be captured on camera. The light passing through the pinhole will reach the detector, which will capture a single image. This process will be repeated several times while changing the direction of the rotating mirrors to take different images in different angles of the specimen sample. Once this process is done, the images will be placed in a computer so that they can be stacked together to form a complete 3D image. Remember, the confocal microscope focuses only on one spot of the specimen to capture specific depth to benefit production of the photo. There are two types of detectors that can capture images in the confocal microscope. The first one is known as the PMT, short for the photomultiplier tube. This detector is used for light detection of weak signals and respond to both photons and electrons. The second detector is called a charge coupled device, or CCD for short, known to be the most commonly used detector in confocal microscopy. Both detectors are sensitive to the light that is being emitted and capture the images rapidly.
Hey everyone, I'm Barry the Bacterial Cell, and I'm here to talk to you about the advantages of confocal fluorescence microscopy over other types of microscopy. When compared to confocal fluorescence microscopy, other wide-field microscopes exhibit a problem when trying to obtain a clear resolution image. Secondary fluorescence emitted from thick specimens obscures a resolution. The solution to this problem is the use of confocal fluorescence microscopy. In this type of microscopy, the two pinhole apertures positioned in confocal positions, hence the name confocal microscopy, serve as a spatial filtering technique that helps eliminate out-of-focus light or glare. Confocal fluorescence microscopy allows for control of depth of field and the ability to collect serial optical sections from thick specimens. This is important when trying to obtain a clear resolution image of a specimen. Confocal fluorescence microscopy allows us to create 3D images of cellular structures. This is extremely important because it gives researchers the ability to examine and observe the cellular processes of specific structures.
Hello muchachos, I'm Gamma Venin, or my friends can call me Mr. AAV, referring to antibody. Remember my name since it will be helpful in the future. I will be helping you understand some of the applications that laser scanning confocal fluorescence brings. These applications are very diverse, and there are new applications of this technology constantly being developed, especially in pharmacologic and therapeutic areas. As you already know, protein binding is crucial for cell survival. Protein can bind to ligands, other proteins, DNA, or barrier surfaces. Many weak interactions are important in cell function and appear to be designed such as by nature. A major challenge for pharmacology is identifying molecular drug targets. Examples: disease-specific proteins, receptors, enzymes, and genes. Laser scanning confocal fluorescence provides imaging applications to study dynamic cellular processes. This digital imaging provides a tool to be able to assist in quantifying special parameters on cells. I hope you haven't forgotten my name yet. Remember? Gamma bending. Thanks to this method, the molecular uptake of gamma bending by cells can be observed. The digital imaging system helps to find and quantify fluorescence emissions. This process can demonstrate the specific protein interactions in the docking and clustering of the molecule on the plasma membrane, internalization of the complex, and triggering of cellular activation. Extra info to take into account is that it is really important to determine what reaction conditions the process can occur. To make it more simple, it is possible to tag an antibody cell with a fluorescence tag and be able to observe through the images its behavior. For example, like to what other cells it binds, or observe how much of the antibody is being expressed at that part of the body. As previously mentioned, laser scanning confocal fluorescence has many applications. I was only able to discuss one real-life example of this technique can be utilized for. Thank you.
Hey there, it's Manny the Microscope again. Thank you so much for watching our presentation. We hope you learned about the overall workings of confocal fluorescence microscopy. Bye!